Hello everybody, my name's Alex Ashby. Tom and the guys at S4S have asked me to do a video for you guys on the immunology of allergic extrinsic asthma. So that's what I'm going to run through today. It's just the immunology, it's not any other parts of the DAPS account, so this isn't a complete guide to asthma. If you've got any questions about anything that I've said, or you think everything that I've said is wrong, then I hope that's not the case, but feel free to email me. My email's up here and I'm sure Tom will attach it to the video. So, the immunology of asthma. It's all about exposures to allergens. The allergen can be house dust mite feces, it can be mould, it can be animal dander, it can be all sorts of things. And there's two arms to the immunology. There's the first exposure to the allergen and there's subsequent exposures. And these two things are distinct. The first exposure leads up to everything up to the priming of the mast cell with the expression of IgE, which is specific to the allergen on the surface of the mast cell. The subsequent exposures are all about mast cell degranulation. So we'll start with the first exposure, I'll quickly recap that, and then we'll talk about the subsequent exposures. So, on this side, I've got the first exposures to the allergen. This is when the patient's inhaled house dust mite feces allergen or something like that, and it goes down into the lung and it meets this cell called the dendritic cell. Now, when this happens, the dendritic cell is a phagocyte and it's an antigen presenting cell. So what happens is the allergen is phagocytosed, it goes inside the dendritic cell and it's processed with MHC2. This is the major histocompatibility complex 2, and it's processed with this, and a short series of amino acids from the allergen are attached to the MHC2 molecule and expressed on the surface of the dendritic cell. So let's draw that here. So this is the MHC2 molecule with a short series of amino acids from the allergen expressed on the surface, like that. Now, the next time this dendritic cell comes across a T helper 2 cell, this allergen bound to MHC2 will bind to the T cell receptor on the Th2 cell and cause activation of the Th2 cell. So let me draw this in now. The MHC2 allergen complex binds the T cell receptor and causes activation of the Th2 cell. Let's draw this like this. This is an activated Th2 cell. Now, activated Th2 cells secrete cytokines, IL-4 and IL-13 most importantly. So they secrete IL-4 and IL-13. Now these cytokines are involved both in the activation of B cells, the other lymphocyte in the blood, to plasma cells, which means that they start secreting immunoglobulin, and they're also involved in the class switch of this immunoglobulin to IgE rather than any other immunoglobulin. So, IL-4 and IL-13 secreted by Th2 cells causes activation of B cells into plasma cells, and because of the class switch, it causes plasma cell to then secrete IgE. IgE is shown as this Y shape. Now, like all immunoglobulins, IgE is made up of two portions. It's got an Fc portion and an Fab portion. The Fc portion is this part here, it's the stick of the Y, and this is the constant portion of the antibody. This is the part that binds to the receptor on the mast cell, which I'll tell you about in a second. The Fab portion, I'll circle here in blue, is the variable region of the IgE molecule. This is the part of the molecule that is specific to the allergen, which was first phagocytosed by the dendritic cell, and this is the variable region of the IgE. So, the now plasma cell secretes IgE because of the class switch from IL-4 and IL-13, and this IgE, which is specific to the allergen, travels around in the blood where it meets mast cells in the lung. Now, these mast cells express the Fc epsilon R1 receptor, which just means that they bind the Fc, the constant portion of the IgE molecule, Epsilon means that they bind IgE rather than any other immunoglobulin, and R1 just means it's the first type of this receptor. There's other types of this receptor. So this IgE molecule meets the mast cells and binds via its Fc portion to the Fc epsilon R1 receptor all over the mast cell like this. I've only drawn it 3 times, but it'd cover this mast cell. Now, this mast cell is now primed. It's ready for the subsequent exposure with the allergen. So if I quickly run through this first exposure again, what's happened is the allergen has been inhaled, it's met a dendritic cell which has phagocytosed it, processed it with MHC2 and expressed it on its surface. The allergen fragment on the MHC2 interacts with the T cell receptor on the T cell, which activates the T cell, which secretes IL-4 and IL-13, which cause B cell activation into plasma cells and the class switch into IgE. The plasma cell produces IgE, which binds via its Fc portion to the mast cells in the lungs to the Fc epsilon R1 receptor, priming the mast cell. I hope that makes sense. Okay, so let's talk now about the subsequent exposures of the allergen. If you remember from the start, this is all about mast cell degranulation and activation. The mast cell from the first exposure is primed, but it's not activated and it hasn't degranulated. So, subsequent exposures, what happens? The allergen again is inhaled because the patient has come across the cat dander or the house dust mite feces again. They inhale the allergen and this time it meets the mast cell in the lung which is expressing this specific IgE molecule. The allergen cross-links 2 of these IgE molecules, which causes activation and degranulation of the mast cell. So now I'm going to talk about what happens when that mast cell degranulates. Okay, so Tom's editing skills have now given me this whiteboard instead of the one behind, and on this I've drawn a graph which shows FEV1, a marker of bronchoconstriction, against time. Now remember, this graph is for the subsequent exposure of patients to the allergen. This is for patients who have already had the first exposure, the mast cells are already primed and they're already expressing the specific IgE on the surface of their mast cells. So here comes along the allergen at time equals 0, the subsequent exposure. It comes across the mast cells and binds specifically to the IgE on those mast cells, causing mast cell degranulation. Now the first thing that happens is these granules fuse with the membrane, releasing lots of histamine specifically. The mast cells also start to produce cystinoleic trienes, but this takes a while longer, and they also start protein synthesis for cytokines, but this takes a lot longer. So, firstly, the histamine causes bronchoconstriction, so the FEV1 declines, but this histamine is quickly degraded. The drop in FEV1 is kept up by a bronchoconstriction by the cystinoleic trienes, which is also being produced. So this is histamine-induced bronchoconstriction, reducing the FEV1, but this is followed by a cystinoleic triene, which I'll put LT, induced drop in the FEV1. This, together, is the early airways response. So it's the early phase of the bronchoconstriction in these patients. What happens then is the cystinoleic trienes are also broken down, and the FEV1 almost returns to normal. But if you remember, also protein synthesis was occurring in these mast cells and they were creating cytokines. Now these cytokines bring in eosinophils, which also release their toxic products and lead to bronchoconstriction. So this causes the late airways response here. So here we've got the early and the late airways responses, but this has been done in a lab where at time equals 0, the patient has been told to inhale the substance that they're known to be allergic to, and then you've seen these 2 airways responses. Now clinically, that wouldn't happen. Patients who come across something they're allergic to, they don't freak out and run away at time equals 0. They might be exposed to the cat dander for a period of time, and this will cause the 2 airways responses to be clinically overlapped, and you won't be able to tell the difference between what bronchoconstriction has come from what. I hope that makes sense. Again, email me if you've got any questions. and thanks for watching.